thank you for the opportunity to show some of our results. I will start by saying that this is work that we have started recently a few months ago, so it's about a year ago. So it's still ongoing, and some of it is preliminary, but I think it's very interesting results. So I will show it here. So just to start, we have a unified research network for this project. That is a collaboration with, I'm from the Sagol Department of Neurology from the Haifa University, and we are collaborating with Technion with 2 labs, and Ben Gurion over the south of Israel, and our clinicians that are working with us are the Emek Medical Center and Sheba. And this is a collaboration and synergy effort. And we have our research aims are starting with drug therapy. So we're testing FDA-approved drugs, and I will show we're testing it on the human and neurons. And these are since they're FDA, they will be easily implemented in the clinic later. And I will show we are planning some gene therapy by messenger RNAs and gene editing and editing. Sorry. So the research tools are the drugs that are available for testing, RNA sequencing and functional analysis to measure the neurons. We have also cellular disease models, which are the brain organoids. This is in my lab, and animal models, which is in a Technion. And together with Avi Schroeder from Technion, we're looking at the way to deliver the drugs, better ways to deliver drugs and gene therapies to the brain by lipid nanoparticles. And later at our collaborations at the Emek Hospital, we will do preclinical trials. So I will show the stuff that we've been doing for the last few months in my lab. So my lab focuses on the iPSCs, induced pluripotent stem cells, derived neurons. So we generally start with some type of either cells from the patient, either blood cells or where we extract the PBMCs, the peripheral mononuclear blood cells, or fibroblasts. And then these are the PBMCs, you can see them here. Then we reprogram them into induced pluripotent stem cells, and then we differentiate them into neuronal cultures and brain organoids. The brain organoids we can also graft into mice brains to allow them for further maturation. So this is a precise disease model in the sense that the neuronal cultures have the exact same genetics as the patient. So it alleviates the need to develop a mouse model for each of the different mutations. And overall, we consider it like a precise model. And again, in the sense that same DNA. We have the organoids and the neurons have the same genetics as the patient. So this is how it looks like. So we start with induced pluripotent stem cells, and then we differentiate them into neural networks, for example. So we can see here some staining that we did in the lab. This is the iPSCs. So we stain for the pluripotency markers. And this is how the embryo bodies look like. And we can, from the embryo bodies, we can either go to do brain organoids or the monolayers. And we have the neural rosettes. We can see some image here. The neural progenitor cells and nestin and PAX6 are specific markers. So most of the cells, once we have the neural progenitor cells, express them. And in the end, we have the neural networks, and we stain for cortical markers such as TBR1 and others. So this is the cohort that we have right now. We have a total of 5 patients. We can see here the mutations in the patients. The red ones are the mutant lines. And for some of them, we have controls which are the first degree relative. So it's almost same genetics or similar genetics. And these are the mutations, and these are the symptoms of the patients and the type of mutations. And overall, for each of these patients, we differentiate the cortical neurons, and we, for the first stage of this project, we are looking at different drugs. So my lab uses a lot of functional analysis. So whole cell patch clamp, it was shown here yesterday a little bit, but generally it was invented in the 70s by Erwin Nair and Bert Suckman, which received the Nobel Prize for it. This is the general circuit. This is the cell, and we have a pipette with an electrode, and then amplifiers that go and get digitized into the computer. and it allows us to see a single ion channels, specific currents, and allows a really thorough investigation of how what are the cellular changes. So we can see here the action potential of the neuron. This is the way the neurons communicate. So, we start with the resting membrane potential. Then there's an abrupt depolarization in the order of 1-2 milliseconds. And it goes back. This is the action potential. And this is how an example recording looked like if we have a voltage clamp. So we clamp the cell at different voltages, and we can see then what type of currents it creates. Or in current clamp, we can see the action potentials. So initially we started with one of the lines, and we saw, we started to differentiate the cells. And when we differentiate the cells, we have overall, we differentiate them approximately for 14 weeks until they're mature enough. But we usually do the experiments throughout the differentiation to see where are the differences that are occurring. And we saw that it was a little bit of a surprise, and I will tell why it wasn't a complete surprise in a few minutes, but we saw that the cortical neurons at this stage, the control neurons, they develop very slowly. And at this stage, they're basically not very excitable. They don't have a lot of sodium currents. We can see here the downward deflection is a sodium current. You can see with the GRIN 2B, it has higher sodium current, and it has more action potentials. And we quantified this, and this was significantly, statistically significant. And also, we're looking even at the connections or excitatory postsynaptic currents rates and amplitudes. And we saw that overall, the GRIN 2B seemed like a more mature neuron. So why was this not a complete surprise? Because about 2 or 3 years ago, we had a study with a patient with an IQ-SEC2 mutation. This is also causing autism, intellectual disability, and severe epilepsy. And these are neurons that we differentiated, and there are around 5 weeks. And we can see here a similar phenotype. The control neurons are still not excitable. They're very young, immature. But the IQ-SEC2 neurons develop much faster. And we had several parameters to show, and it was significant. And okay, this was early stage. This is 5 weeks. But when we keep differentiating them, what happens is that the phenotypes flip. Now the IQ-SEC2 is less excitable than the control. We can see here that was significant, but if we keep differentiating, it becomes more significant. But also, and importantly, the IQ-SEC2 neurons, so this is the 5 weeks, they had more synaptic activity than the controls. We can see these lines here. They're the synaptic activity. But when we go to 11 weeks, they then didn't develop much with the synaptic activity, but the control developed a lot. Now they have a lot of synaptic activity. And so at the late stage, when the cells are mature, actually the mutant lines have less synaptic activity. So, and then we had a bunch more mutations that we did, and we saw this, we published this, called it an early maturation of ASD. This is the GRIN 2B, which I showed. We had a patient with a 7-dupe. It's a duplication in chromosome 7 in the genome. SLC1A4, a UBTF, SHANK3, and the IQ-SEC2 all have similar phenotype of this really early maturation. Okay. So, and then now we're going back to the GRIN 2B. We're seeing that they have phenotypes that are similar to other autism lines that we have. And now we have the iPSCs, we're differentiating them into cortical neurons, and we started to apply mostly FDA-approved drugs at this time point, right when we're starting the differentiation. So then we keep feeding the neurons constantly with the treatment. So these are the drugs that we use. Some of them I will show. So L-serine, D-serine, and pregnenolone sulfate, you'll see in the slides. We are also looking at other drugs. Drug 1 was developed at the Tel Aviv University for autism, so we're trying that one. And these 3 drugs have been shown in the literature, drug 2, 3, 4, to increase GRIN 2B expression. Drug 5 is a type of antibiotics that reduces neuroinflammation. And drug 6, which is developed for autism in Shaare Zedek, is another hospital in Israel. So importantly, these are FDA-approved drugs that can be repurposed quickly and for treatment in the clinic. So now we have started to differentiate the neurons, and we're looking at young neurons and old neurons. These are young neurons at 4th week of differentiation, so approximately 30 days or a little less. And we can see this hyperexcitability. Now this is the total evoked action potential, it's the measure of the excitability. We can see the untreated cortical neurons from the patient are much more excitable than the controls. And we see the different drugs, for example, L-serine, that reduces this early hyperexcitability. It makes them more similar to the controls. And how does this look like in the recording? So these are evoked action potentials. We can see the control has some action potentials, but not so much. This is the untreated, and this is with 2 types of drugs. We can see they slow down this hyperexcitability. These are example recordings we have, from each cell. We have usually when we get to publication stage, we have dozens of cells from each patient. Here we have approximately 20, I think, but we're still acquiring data. Some of it is still statistically significant, but these are examples. So it's an example of each cell, but we have many cells of these recordings. And if we look at the sodium currents, for example, so the sodium currents is highly correlated with the excitability of the neuron. We can see here the control neuron. It's not neurons, it's neurons. So it's a average over, you see the N over here. Some of it is still rather low, like 9, but some are higher. But we're keeping on acquiring data. And so we have the untreated, which we can see how much it's very much increased, the sodium currents. And we can see that the different drugs reduce the sodium current. And this is the examples, how it looks like. So this is a control. Again, the downward deflection, this downward towards the minus sign is the sodium current, and this is the untreated, has bigger sodium current. And with the treatment, they reduce this current. So now we're looking at a mature neuron. So this is a 10th week from differentiation. You can see the ends are still low, but we are keeping on acquiring data. So here, actually the cells become hypoexcitable. So we can see, for example, the untreated compared to the control. Now they're much less excitable. And some of the drugs, L-serine, for example, is significant even with just N equals 9. But some of the drugs increase this excitability. And this is how it looks like. The control neurons now produce many more action potentials, but the untreated neurons become hypoexcitable. They don't have much action potentials. And with the L-serine, we can see that they're more close to the controls. And when looking at the sodium currents, which is important, it's correlated with the excitability. We can see now the control is now higher, while the untreated is now lower. It flipped, similar to what we saw with IQ-SIC2. And we can see that, how the drugs affect. Most of the drugs increase the sodium currents. And this is how it looks like. Examples, so we have the control. We can see the sodium currents. The untreated sodium currents are smaller. And this is L-serine and drug 4, which was also shown to be quite effective. I talked a little bit about the synaptic activity, but just a quick slide. What is the synaptic activity? So we have the synaptic cleft, and while a neurotransmitter is released, ion channels open up and allow ion currents into the cell. And these are the currents that we see, these lines going down. So these are actually synaptic currents. So these are currents from other cells. So it means that the other neurons are sending. It could be either they're spiking the other neurons, or it can be spontaneous also. But overall, when we have more, and if they have a higher amplitude, it means there's more synapses and they're more efficient. So looking at the excitatory postsynaptic currents, we can see here an example of a control. They have a lot of synaptic activity, while the untreated ones have less. But then we have some drugs that increase. Again, we're repeating, the ends are still low. For example, with the amplitude, we see, this drug 4 gives the most change, but I think that the other drugs, once we repeat the experiments, will also show some of them, at least, will be more significant. So now we're going to another. So this was one patient This is another patient, the 3380, with a nonsense mutation. And we're repeating this. So for some of them, we have less drugs, but we can see this phenotype, which is repeating. When they're young, they're hyperexcitable. Some of the drugs reduce this hyperexcitability, young hyperexcitability. And this is the way it looks like in the examples. We can see the controls and the untreated, which are much more excitable. And some of the drugs reduce this hyperexcitability. Similarly, with the sodium currents, we see, again, the control is reduced compared to the patient. And some of the drugs reduce these sodium currents to make them more like the controls. And these are some examples: control, untreated, higher sodium currents, L-serine, and drug 1 treated, which reduce these sodium currents. Now we're looking at the mature neurons. With the mature neurons, again, we see now this flip in the phenotypes. The control neurons, now they were very less excitable because they were young, immature. Now they have matured. They have many action potentials now. This is the average of around 100 action potentials in our measurements. And while the untreated patient, or neurons from the patient, are much more reduced in excitability. And some of these drugs, for example, L-serine, increase this excitability. And this is examples. We see the control now have a lot of action potentials, while the untreated now have reduced a number of action potentials, but L-serine and drug 2 increase this. Looking at the sodium currents, again, here the phenotypes flip. The untreated patient is less than the control, and some of these drugs increase these sodium currents. And these are some examples. I'll not repeat. And looking, I'm sorry, this is the, okay, now we have another patient. This is a patient with a nonsense mutation. And here we have also young neurons, which are basically, these are not as hyperexcitable, but we're repeating this also. We think it will change a little bit, but they are hyperexcitable. And some of the drugs reduce this hyperexcitability. And we can see the examples of the control and the untreated, and L-serine and drug 5 treated. Sodium currents, similar to before, you have the control, which are lower, and the untreated, which are higher, and some of the drugs reduce these hyperexcitability, this increased sodium currents. And some example of the control, untreated, and treated, which reduce the sodium currents. Looking at the mature neurons, we see, again, this reduction and this flip. Now the control neurons become more excitable as they mature, but the untreated, they generally do not develop as much. They're less excitable. Here, drug 1 was the best one to increase this excitability. And we can see the examples of a control and untreated and drug 1. And similarly, the sodium currents here, there was not a significant, very big change, but there was some change. And some of these drugs also made them more like the controls. And these are some examples of the control, untreated, and with drug treatments that increase these sodium currents. And we have also the excitatory postsynaptic currents. Both the frequency and the amplitude are reduced. So the ends here are still, so the excitatory postsynaptic currents, usually the frequency and also the amplitude, it's very variable. So sometimes we need a higher ends. But so we need to repeat the experiments. But we see from the examples, that most of the untreated cells look like that. And by the other drugs, they have some cells that have synaptic activities. Well, others don't. But we are repeating this. Also, we're looking at calcium imaging, doing a network analysis. So this is how we have algorithms for, we're looking at the neurons that fire together. We have algorithms first to identify which cells are active, and it marks, the algorithm marks which cells have activity. And then we cluster them into groups that fire together, that are synchronized. So we can see it here with the colors. And overall, usually what we see, this is an image that we see, it's a video that we run. We see the calcium imaging, we apply calcium indicators onto the cells. And when they're active, they have a fluorescence. So we see an increase in this fluorescence level. And by measuring which cells fire together, we can know their connectivity. So we're looking at a network analysis, and we can see this is an example of a network analysis that we did. So each dot here is a neuron, and we can see the ones that are close together. It means that they're very connected, and then they also have connections to these clusters. And this is a control neuron, and this is the green to be the 7843 line at 10th week. So at 10th week, if you remember, the cells are less excitable and they had less synaptic activity. So when we map out the connectivity, we can see that there is a reduction in the connectivity of the neurons in the neural network. But when we treat with the L-serine, we can see that it doesn't become like the control, but it's much more connected. This is still, so we have a ton of recordings that we're still analyzing them, but this is something that we are gaining out statistics, but we see that, some of the drugs, I think this is a very important measure, the connectivity of the neurons, and some of the drugs really increase the connectivity in the mutant lines, which are much less connected. So just to summarize this part, we're using human organoids and neurons as a platform for personalized medicine. So for each patient, we are doing this procedure of making iPSCs, making cortical neurons, and testing. We have a total of approximately 15 drugs that we will be testing. And then we can see which of the drugs is suited best for each specific patient. And this method allows to test drugs quickly and specifically for each patient. So this is a project that we just started to do with the Schroeder lab at Technion, Avi Schroeder. So they're developing lipid nanoparticles, and basically we together with them, we want to take, they will incorporate drugs or wild-type messenger RNA into lipid nanoparticles. Right now, we're applying them to the dish, but at the later stages, we'll use mouse models and later, the clinical trials. This was just a very preliminary test to show we incorporated M-cherry. So Avi Schroeder incorporated M-cherry into their lipid nanoparticles, and we put it on the human neurons, and we can see that after 24 hours, many of the cells express this mRNA, which means that we can put another mRNA like the wild type. So we have some results from there, but it's very preliminary. I'm not showing, but I will show we just published a study for Parkinson's disease, so it was led by Avi Schroeder, but we helped with a human model. It's brain-targeted liposomes that were loaded with antibodies against alpha-synuclein to reduce alpha-synuclein aggregation they had mice models and also in the human neurons. So basically, in Parkinson's disease, we have the affected areas of substantia nigra pars compacta, and the neurons there, the dopaminergic neurons, tend to have aggregates of the alpha-synuclein protein, which misfolds and forms these fibrils. And the approach was to take antibodies for alpha-synuclein, incorporated there its liposome, not the lipid nanoparticles, but we now have so it's Avi, but we're now developing a better method. And to put it onto mice and to human models with alpha-synuclein aggregation. So we have the mice models for Parkinson's disease, and for us, we have dopaminergic neurons that were derived from Parkinson's disease patients, in a similar manner to what I showed. And so we've incorporated the alpha-synuclein antibody into the liposomes over the Schroeder lab. They are specific. They can specifically enter by the transferrin receptors in the brain, enter and into the brain and reach the neurons. And so this is the entire scheme. We have the liposome with the antibodies that enter the brain, reaches the neurons, and reduces the aggregation of alpha-synuclein. And this is just some staining with human neurons, where we show, the neurons uptake of the liposomes. So summarize the second part, which is just starting, but this is we know that all not all therapeutics reach the brain because of the BBB, but the LNPs allow for targeted delivery, and our pilot shows that this is possible. And we have started to work with the ASD cortical neurons for the GRIN2B mutations and the lipid nanoparticles for both drug delivery, as well as in the later stages, we hope to deliver wild-type messenger RNA. So with this, I would like to thank, these are my students who worked on this project. We have our collaborators over at Technion, Professor Avi Schroeder. We have our collaborators at Emek Hospital, Dr. Tali Garin-Shkolnik and Professor Ronen Spiegel. Our collaborators at Tel Aviv University, Professor Ilana Gozes and Professor Adi Aran from Share Tzedek. And the GRIN2B donors and participants. Importantly, this project is being funded by the To Be Cured, which is a patient organization that funds this collaboration, which is really oriented to finding therapeutics. And thank you all for listening.